So next up, we have Dr. Petra Kluger, who's a professor of tissue engineering and biofabrication at Reutlingen University, and she was before also the vice president of strategic development and the vice president of research there as well, if I'm correct. And a fun fact about Petra is that actually she presented a keynote presentation at this conference in 2018 and wasn't working on cultured fat or cultured meat at the time, was just working on adipogenesis and fat tissue engineering with regards to other applications, and was so inspired by that meeting that she actually created a group in her own lab to work on cultured meat and cultured fat, and now she's here to give a presentation to show what amazing work that group has done in this time period. So please welcome Petra.
Thank you very much for this kind introduction, and maybe I can add, after the conference in 2018, we did not have money, as most research groups, to start what we want to do, but we got here a project started after the conference, and these are the results ending now of this project. So Annemarie did a lot of work here, and a lot of other people too that are sitting in the audience. She's not here, but I will present the whole data. So oh, there's a screen, great. So we have learned in the talk before a lot about fat tissue engineering, and it was really a great talk, and thank you very much. So I can skip 2 slides and save time. Because we all know, okay, fat is needed in cultured meat, and we have learned already that we have the differentiation, and maybe just, you know, I'm a professor, so you maybe keep in mind we have to learn something. So what we see in the stainings are the lipids inside the vacuoles. I know we have some people in the audience that are not from the biological research area. And just keep in mind, if you see the green or yellow stainings, or you have seen, these are the lipids. And another important protein is here at the lipid membrane. This is perilipin A. I will come back to this later. So our aim 3 years ago, where maybe this is part of the aims we had, because, you know, if you start in a really new research area, sometimes you are too, you look too much in the future and you come back to the basis, because cell isolation with the animal cells is quite different than working with human cells, we have learned. Janis can tell you some stories about this later on. But what I want to present here is that we had the aim to see whether we can achieve from bovine adipose-derived stem cells, not the FABs we have heard before, but the other cells, whether we can work with them, because we have a lot of experience with them in the human system. And we also struggled with the media. We didn't have a serum-free media, but we worked antibiotics-free. This is not that easy at all. And we skipped some parts too, and we really saw similar results. This is really nice, because then it seems that it works for other groups too. And the idea was to see whether our bASCs form spheroids, and we looked at in static and dynamic culture conditions with our serum-free media, and thereafter we wanted to use these spheroids as building blocks to build up larger constructs, and we printed them, and we did, again, what you have seen before, this fatty acid analysis. And so I jump in directly. Maybe something different than most of the other groups are working at is that we really use waste products, so from a butcher. And we have really 2-year-old animals, or the rest of the animals that the butcher does not need. And we started to get them isolated. As I mentioned, this was a long work, but at the end it was quite fine. And after the first isolation step, when we passage the cells, then we can skip antibiotics. So quite similar what you have shown. We worked with the differentiation media, and we have similar results, because we can skip the IBMX and we can skip the indomethacin. We still have the rosiglitazone and the DMEM, and of course we are working with FCS, but we have seen a lot of interesting work how to reduce it. This is focus of other research topics we are focusing on, and we have the rosiglitazone and the bovine insulin inside. But this works fine for our adult bovine ASCs. And if you want to learn more about our serum-free approaches, you can visit Tobias at his poster in the poster session. So these are our spheroids we produce out of the bovine ASCs. In the static culture, we started with different cell numbers, and interestingly, all worked quite fine. And if you look inside our spheroids, these are cryosectioned ones. We do not see any cell death markers inside. And for us, mainly important was how to differentiate the cells. And if we made the stainings, you remember the yellow dots, these are the lipids in our staining. We can see that by using our differentiation media, we have really nice differentiation inside, and we can see the perilipin, this is the green staining for the static culture. And of course, static culture was just the beginning, because then we tried to produce our spheroids in a dynamic culture. These were just shaking Petri dishes, so simple protocol, but I think an important step to get them into the bioreactor we are working at the moment on. And again, you see, of course, the spheroids are different shaped, because it's not that you put 10,000 cells into one well plate, but it's just, for example, 250,000 cells that we have per milliliter, and then this spheroid formed automatically in whatever size. And we shake them for 2 to 3 days that they find each other to adhere. And then we started a differentiation. You see again, we have stained the cell death markers, and there are no cell death signals inside the spheroids. And again, in the shaking culture, we see here the accumulated lipids for our adult bASC spheroids, and the perilipin again. So let me see, I forgot nothing. Great, because if you want to know more about spheroids, you can visit Anna's poster. So you see we are here with the whole fat or meat group, a little bit smaller than at Mosa Meat, but increasing since 2018 tremendously. And yeah, I want to show you now what we are doing with our spheroids at the moment. So interestingly, you know, I'm coming from the biomaterial part and biomedical part, and we like to print. You do not always have to print, but printing is an interesting technology, and it's just one method how to use these spheroids or whatever kind of aggregates for producing larger constructs. And we have, if you want to learn more about printing, yes, Yannis is here too, and he has a talk tomorrow, and this is the last one I've mentioned, Simon, he has a poster, so this is the whole group. And what I want to show you today is an edible bioink. This is gellan gum, widely used, food grade, available, and we have developed this material as a bioink, and a bioink means the material and the cells inside. And we do not only incorporate the cells, but also the spheroids, and then printed it with our bioX printer. And maybe when we start with this picture, you can see here the spheroids. These are the white dots, yeah? So here is only, so the first picture is only the bioink, or only the material without the spheroids. And you see if you include the spheroids, you see these little dots. And like most people in bioprinting, we print these stupid grids because of diffusion limitation, but this works quite well. And when we look inside, oh, I forgot to mention, we introduced differentiated spheroids. This means using them directly to produce a product, not that tasty like your fancy burger, but what we can see is that the cells are viable inside after the printing process, and that we have still the lipids incorporated. And lastly, you have seen quite similar pictures before, but maybe just again the fatty acid analysis. This is really interesting, because what we gain. So you remember we start with this bovine fat tissue, and this is what we achieve. These are our differentiated spheroids. You see they have this yellow color too. And maybe this was the question from here. We have analyzed in comparison the non-differentiated spheroids and the differentiated spheroids, and done this fatty acid analysis with a GCMS. Does not matter, but it was in collaboration with the University of Hohenheim. They have a big core facility, normally working with kilograms and tons of whatever kind of food. They had to scale down their techniques, and what we can see here, and maybe we mainly look on this first picture. These are unpublished work, to let you know. You can keep your fingers crossed that it's published soon. And we have here our control spheroids. These are the non-differentiated, and we saw this shift. You mentioned it before. You can really see it during the analysis that we have a shift in the fatty acid composition. And maybe I have to mention we have the saturated fatty acids, we have monounsaturated and polyunsaturated fatty acids seen here. So we see especially the shift in the monounsaturated ones. And if you compare it to the fat tissue, we see that our differentiated spheroids look quite similar to the native tissue. But of course, there are some differences. But for us, the most important was to see this shift that we have seen in the stainings before, but for the lipid or for the fatty acid composition, it was interesting to see it. So, oh wow, I've spoken too much, but maybe we have more time now for questions, because there are 4 minutes left. I just sum up. We have shown that we can produce out of adult bovine adipose-derived stem cells that we gained from slaughter waste, these spheroids, and that we can differentiate them with our modified differentiation media. And I've got a lot of insight the last days how to improve it further. We have done this fatty acid analysis and saw here the differentiation by the shift in the composition. And we use them as building blocks for further processing. Bioprinting is one technique. There are many other techniques how to use them. And next would be for us, or what we are working at the moment on, is the serum-free media and the optimization of the proliferation and upscaling and the evaluation and development of further biofabrication techniques and materials for that. So with this, I want to thank you all. This is the group, the whole group. We are still working in the biomedical field, but to be honest, the biggest part is the cultured meat group. And I especially want to thank the Avena Foundation that I met here in 2018, and we got this great project that will end in end of October, and this was really a big chance to get started into this area. So thanks for your attention.
Thanks so much. Do we have some questions? Or from online? Okay.
Hello. Yeah, nice talk. This is really a question to you, and also for the last talk. You talk about fat, but what type of fat are you making? Is it white fat, brown fat? Obviously, or beige, because obviously rose because rose tends to brown fat. Yeah.
No, it's white one.
Okay, thank you.
Can we toss it over here, because I think we've been. John had his hand up the last session as well. Thanks.
Thank you. Thank you for your talk, Dr. Kluger. I was wondering if you have what looks like differentiation of the fat in suspension. Do they end up floating, and then is that would that be an issue for large scale?
Yes, interestingly, if you differentiate them for a longer time, you mentioned it before, they start to float because they have these lipids. Maybe not all of you know the mature adipocytes, if they are in single cell, they float on top. And of course, it is interesting to harvest them later on.
Anybody else have a question? Or does Mark has one.
Oh.
Finally get to do this. So if you look at the wonderful talk, by the way. If you look at the fatty acid profile in the undifferentiated ones, it actually looks pretty good, right? So because it has, polyunsaturated fatty acids, who doesn't want that? So why do you still differentiate?
Yeah, interestingly, we did not know what we will get, so we were really astonished that it is that fine, to be honest, for the undifferentiated ones. But I think, now we can start to modulate, maybe like you plan to do, And yeah, maybe for the taste, it's necessary to shift here anything. I'm not sure about how to go on, but we do not have the lipids, that much lipids in the undifferentiated ones, so we need it maybe for the texture or whatever.
Oh, so it's more the amount, because the cells morphologically look very different. So it's more the amount than the fatty acid composition.
Yeah.
That sort of drives you to do the differentiation.
Yeah, actually, we did it at the end, so
Right.
yeah, it looks for both. I think for both spheroid, it looks quite well. So maybe we do not have to achieve the perfect differentiation at the end. I often think this for muscle too, to be honest. Yeah, because maybe something in between would be fine enough for taste.
Yeah, we'll have a panel discussion about that tomorrow.
Okay, great.
Oh, well, we're perfect for timing. You have 4 seconds left, so thank you so much.